Paul, can you give me the codon for leucine? The first codon for leucine from the table that I have shared with you.
Sir, in the chat box, there is only one message from Monica. Not, I couldn't get your file, sir.
How is that possible? No, right above Monica's message, there is my message. I am pasting it again.
Yes, sir, there is a link.
Open that link. Do you see that now?
Sir, sorry, sir. No, sir, I couldn't get that file. It is possible to, you can send it again. Because I'm seeing only one message, sir. The recording is not on from Monica.
Okay. So someone said that he has seen the message. Who is that person?
Sir, I have seen it, but I am doing from mobile, sir.
I need someone who is working with a laptop to open that link.
I'm using laptop, but it is not also visible from here.
Yes, sir, there's nothing on the chat box.
How is that possible?
Can you check who did you send it to?
Okay. It is...
Sir, I'm using Google and I can see that.
I know, I made the mistake. Okay, it is gone now. Can you see now?
Visible now, yeah.
Yes, sir, it is.
Yes, sir. Okay. So I need someone to open that table because I will need your help. Okay, done?
Yes, sir.
Okay. Yes. So give me the first codon for leucine. You just say it, I will be able to write. UUA. Okay. Arginine.
A.
Huh? I didn't get it. Arginine.
CGU.
Huh?
CGU.
Okay.
Yes.
CGU. Okay. Threonine.
ACU.
ACU. Good enough. Okay. Stop codon.
UGA.
Sorry. In some parts of the screen, it does not work. Okay. So, fine. Now, this is DNA, okay? Please never write DNA or RNA without 5 prime, 3 prime, okay?
Yes, sir.
And I made a mistake here.
Yes.
Sorry, this particular part of the screen, for whatever reason, it doesn't work. So those who are relatively unfamiliar with biology, you see that just because I have written the top strand, I can automatically write the bottom strand. Okay, let us say this is a double-stranded DNA. Okay, and somewhere over here there is a promoter. Okay, we will study promoter in detail right now. The only thing that we need to understand is promoter is where the enzyme that polymerizes ribonucleotides with direction from DNA, that is the enzyme called RNA polymerase, lands. Okay, so this is where it lands. It's a very large protein complex, and it starts to transcribe somewhere approximately 10 to 25 bases downstream. Okay, and if the PhD interview is anything to go by, let me inform you in no uncertain terms that transcription does not begin from here. Okay, it is absolutely untrue. Transcription does not begin from ATG. Okay, many of you have that misconception. At least during interview you made that kind of statement. That is not true. Okay, so now the RNA is being made. Okay, and so when the RNA is made, there instead of T, they all become U. Okay. So of the top two strands, the polymerase uses this strand to read, and it moves in this direction, and the first nucleotide added is its complementary, that is A. The next nucleotide added is this complementary, that is T or U, and so on. Therefore, the transcription happens from 5' to 3' direction, that is this, but it reads this one, okay, which is called the template strand, and this is the sense strand. Anyway, so now let us understand how the reading frame is set. And I am teaching you today. You will need to remember it when I will be actually teaching you translation, maybe 4 or 5 weeks from now. So do not ascribe any special characteristic to ribosomes. Ribosomes are like any biological molecule. They are not smart molecules in that sense. They do not take any decision. They act by default. Okay. So what is it that they do? So if it is an eukaryotic system we are talking about, eukaryotic mRNA, messenger RNA, they will have a structure called the cap structure. Okay, it's a modified nucleotide, cap structure. And in eukaryotes, ribosome lands on the cap structure. Okay, and then it crawls, okay, from 5' to 3' direction. And there, when it comes to the AUG, there, up to that point, it is just the small ribosomal subunit. Just hold on a minute, please. So there, the small ribosomal subunit lands, and only there, the large ribosomal subunit comes and joins. Okay. Once that happens, then it starts to crawl. Now, the issue is that what I'm trying to impress upon you is that the ribosome has no choice. The first AUG that it will encounter after being recruited onto the mRNA, it will have to start translation right from there. It has absolutely no choice. This region is referred to as 5' UTR, untranslated region. Now, for majority of the mRNAs, in this region, there will be no AUG, because if there is an AUG, translation will start from there. Now, the fun part is that the moment it encounters an AUG, then from that point onwards, ribosome starts to count in groups of 3. Okay? Like this. So it has no option but to incorporate a methionine here, a leucine here, an arginine here, threonine here, and here it is stop. Okay, it has no option. In prokaryotes, it is slightly different. In prokaryotes, what happens is that in the middle of the RNA, they will have a sequence called Shine-Dalgarno sequence or ribosomal binding site. Now, wherever the ribosome sees that sequence, there, so the Shine-Dalgarno sequence has sequence complementarity with the small ribosomal subunit. One of the RNAs of small ribosomal RNA, that is the 16S ribosomal RNA. So the small ribosomal subunit will land on there. So in eukaryotes, the small ribosomal RNA lands onto the end, that is the cap structure, it is the end. But in prokaryotes, it lands onto wherever it sees a ribosome binding site. And after that, it starts to crawl and it reaches AUG and then everything remains the same. Okay? So that's how translation happens. Now, will someone please give me another codon for leucine other than UUA?
CUU.
Huh?
CUU.
I don't need that one. I need starting with UU.
UUG.
UUG. Very good. Okay. So, if for some reason there is a mutation that this A is changed into a G, then the amino acid that will be produced is still leucine. I will explain it later, but for those who are not very familiar with molecular biology, you should know that this set of 3 nucleotides, which is called codon, they have degeneracy. Let me explain it to you. So there are only 4 nucleotides, right? And everybody knew that there are 20 amino acids. So when scientists were thinking about it, that how could 4 nucleotide code for 20 different amino acids, they realized that both permutation and combination will be in play. 1 nucleotide coding for 1 amino acid is not possible because then only 4 amino acids will be coded. 2 nucleotides coding for 2 nucleotides coding for amino acids is also not possible because the first location and the second location. The first location can be either of A, T, G, C. So it is any one of 4. The second location will also be any one of 4. At this rate, you can maximally make 16 different amino acids. So you cannot account for 20 different amino acids. If you take 4, that all 4 nucleotides are needed, like the codon is 4 nucleotide long, then the first position can be filled in any one of the 4 manner. Second one will be any one of the 4 manner. Third one will be any one of the 4. Fourth one will be any one of the 4. You will get 256 varieties of combinations, permutation and like combinations. So that is too much to account for only 20 amino acids. Therefore, and of course many other experiments were there. So scientists figured out that a codon is only 3 nucleotide long, and this will account for 64 different codons. Of these 64 different codons, 3 codons signify stop. Okay, they do not indicate any amino acid. So 61 other, they will all indicate one amino acid or the other. So you can imagine that there are 20 amino acids and 61 codons to choose from. Therefore, for certain amino acids, there are multiple codons. Like say, for methionine, there is only one codon, that is AUG in RNA, and in DNA, it is ATG. Okay. But leucine, on the other hand, it has 6 different codons. If you look at the table that I have shared the link for, you will find there are 6 different codons for leucine. Arginine also have multiple codons. Only methionine has 1 codon as far as I remember. So now, once you have taken a good look at the table, while I am teaching, you please keep your eye on the table. That way you will understand it better. So now if for some reason the A here is mutated to a G, the amino acid still remains leucine. It does not change. And because it does not change, this is a silent mutation. Okay. A silent mutation is typically in the reading frame, in the ORF, but does not result in change of amino acid sequence. So it is silent because you did not even realize that there has been a mutation. So that's called silent mutation. It is a different matter altogether when you will read more sophisticated literature. There are examples where silent mutations can also be deleterious, like bad. But generally speaking, silent mutations are not harmful. Okay, so you understood silent mutation. Okay, but imagine that this one did not change. Rather, there is a mutation because of which this U is changed into a G. Now what happens? It cannot incorporate a leucine any longer because suddenly this is synonymous to stop. So this particular codon makes no sense, meaning it does not indicate any amino acid. Therefore, these kind of mutations are called nonsense mutation. Okay, so a nonsense mutation is when a amino acid coding codon is changed into a stop codon because of a mutation. That is called a nonsense mutation. Now, would someone please tell me what is the amino acid corresponding to AUA?
It is isoleucine.
Isoleucine. So if the U is mutated to, like this T is mutated to an A, then what you have is your amino acid leucine is changed into a different amino acid called isoleucine. Now that is where the sense changed. It is not nonsense. It is still a sense, but it is a wrong kind of sense. That's why it is called missense mutation. Okay? Now we will remember this for the time being. Okay, would someone please tell me what is CUA?
CUA is leucine.
Huh?
Leucine.
Okay, GUA?
Valine.
Okay, so I will leave that alone. इसको leucine ही छोड़ देते हैं. Here, please tell me what is GGU?
Sir, glycine.
Glycine.
Okay. So if there is a mutation here, this arginine will no longer be coded. It will now become glycine. Okay. So it is still a single base change, but the change is now arginine to glycine. Now, those of you who have studied amino acids, it is there in your 12th standard book as well, that both leucine and isoleucine are hydrophobic bulky amino acids. So a change like leucine to isoleucine may not be very deleterious, may not be very bad for the cell. So these kind of mutations are called conservative mutation. Okay, that is, the sense is changed. It is a missense mutation, all right, but it is not that bad. It is a conservative mutation. The property of the amino acid remains more or less the same. But this is not a conservative mutation. Here you are replacing a charged bulky amino acid with the tiniest uncharged amino acid that can have serious consequences on the folding of the protein and or its interaction with other proteins. So just hold on a minute. Huh? I have a call. Just hold on a minute. Hello? I'm teaching my class. So that's what it is, okay? So now tell me if any of you have any question. Piyusha किधर गई? Did you understand?
Yes, sir. I understood the change of the codons and also looking up the table. But primarily, when we were designing the strain from 5' to 3', how did we make that change? That I did not really understand.
Oh, that I made it up.
Okay.
You can write any sequence. I just made it up because I know some of the popular examples which will be easier for me to deal with. So I just made it up. Okay. Anything else?
No, sir, no.
Okay. So the class, anybody has any question?
Hello, sir.
Yes, Kuna?
Sir, in prokaryotes, we have the RBS ribosomal binding sites.
Mhm.
In eukaryote, what is the site it's called? And
It is not a sequence. It is a modified nucleotide. It's a methyl guanosine residue with phosphate group. It's called the cap structure, CAP.
sir, the capping is not available in prokaryote mRNA.
No, the capping is not available in prokaryotic mRNA.
Sir, if we design for a for a DNA, if you design for an proteins for like large scale production of some protein like insulin or other, if you make it in eukaryotes, then we have to give some signal sequence in our primer for like capping, or it will automatic added to the like mRNA transcribed mRNA.
A very good question. Excellent question. So when you are using a eukaryotic host, okay,
Yes, sir.
it can be insect cell line, it can be mammalian cell line, it can be yeast cell, then that host has a nucleus.
Yes, sir.
Therefore, your DNA will be transcribed within the nucleus.
Yes, sir.
And eukaryotic nucleus automatically will cap the 5' end and will put a poly A tail on the 3' end. These are both called post-transcriptional modification. I will teach it later in the course.
Okay. Yes, sir.
So it is automatic.
Okay. Yes. Thank you.
Hello, sir.
Yeah.
So I have a question.
Okay, Akanksha, go ahead. Ayush, I will come to you.
Okay.
Yeah. Okay.
Akanksha.
Yeah, so like you said, there is this cap structure, so the ribosome like it binds there, right? Like from the
Akanksha, your connection right now is very bad. I cannot hear you at all.
like is it audible now?
Slightly better.
Okay, so what I was asking is, in eukaryotes, there is a cap structure where the small subunit attaches, right? So
Yes.
when it comes to prokaryotes, you mentioned that there are ribosome binding sites. So like the ribosome like recognizes those sites. So is it possible that in a prokaryotic mRNA there are multiple like ribosome binding sites, and how does the ribosome decide like where to start like transcribing from, like translating from? Sorry.
Very good question. I will answer it a little later. I will answer it. Ayush, what was your question?
Yes, sir. So my question is if we are cloning a eukaryotic gene into a prokaryote for its overexpression, take an example for insulin. If we are cloning that gene in a prokaryote, so will we engineer the gene or the chunk of the DNA with ribosome binding site in that? Because in prokaryotes there must be no protein molecule or the complex that will bind to the 5' cap of the mRNA. I mean, I suppose you got the question.
Very good question. Both are very, very good questions. What is your basic degree, Ayush?
Sir, a master's in genetics.
Okay.
MSc in genetics.
Was molecular biology taught to you at your earlier classes, courses?
Yes, sir. Yes, sir. Yes, sir.
How about you, Akanksha?
Sir, my background is in biotechnology and MSc in biotechnology.
And BSc?
In botany.
Okay. So, okay, I will answer the question. Okay. Give me a minute. Will someone please open an image of lac operon? Okay,
Sir, any
let me do it.
any
Let me
okay, sir. Okay, sir.
let me do it, because you would not know what I am looking for. Just a minute. I'm trying to find the best possible diagram here. Another 2 minutes, huh? Okay, can you see my screen? Hello, can you see my screen?
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
Okay, I will not get into all the detail, but you see, this is the promoter. Okay, this is prokaryotes. Lac operon is there only in prokaryotes. This is the promoter, and we don't need to understand what is operator right now. You see there are 3 arrows. Each arrow is one protein coding region. The concept of ORF is actually a very prokaryotic concept. In eukaryote, we did not need to have this concept. You will understand in a while why. Now, so what it means is that translation will start here and will fall off here. Another translation will start here and, start here and will fall off here. Another translation will start here and fall off here. So these are 3 independent translation events. Why? Because if this is your lac Z, then this is the start, this is the stop, then this is your lac Y, of which this is the start, this is the stop. In between this stop and this start, there will be another ribosome binding site. There will be one ribosome binding site here. And there will be another ribosome binding site for the before the start AUG of lac A. Did you get it, Akanksha? That
Yes.
the ribosome binding sequence is fairly long. If I am not mistaken, it is like 16 to 18 nucleotides long. So the probability of that occurring randomly is very low, right? 1 by 4 to the power 18. Let us assume it is 18 nucleotide long. So exactly that same sequence being there, the probability is once in every 4 to the power 18 bases. So it is not so frequent. But to ensure that all the 3 proteins are translated, you have to have a ribosome binding site in front of each of the start codons. Why? Because the ribosome starts to scan, it comes to this start codon, then the larger subunit joins to it, it scans, then it is the stop, it sees the stop codon and it falls off. There is no way it can go through. So it cannot start again translating from here. For translation to start here, again the small subunit will have to come here, scan this much, join with large subunit, go, and here again it will fall off. So if I did not have the picture readily available, so I request that you take a look at it yourself. Tryptophan is an amino acid that is also made in yeast, right? Or in your cells. And there also you need 5 enzymes in the pathway to make tryptophan. In bacteria, there is an operon. What is an operon? An operon is where you have only one promoter. Okay, as it is very clearly shown here, RNA polymerase binds here. So there is only one event of recruitment of RNA polymerase, and then it moves in this direction. So as you can see, only one mRNA is made. And from that one mRNA, because of the presence of multiple ribosome binding sites, you will make 5 different proteins. Do you think that is possible in eukaryotes? Like, do you think in yeast it will be like this?
No.
Why not?
Because of the absence of multiple ribosome binding site in eukaryotes.
Absolutely. Therefore, the operon concept does not even exist when we are talking about eukaryotes. Because the difference is here, listen to me very carefully, that in prokaryotes, when it comes to transcription, making the RNA, there is only one event and you get an mRNA, messenger RNA with multiple warfs. So one transcription event, but multiple translation events from the same mRNA. While in eukaryotes, you will have on different chromosomes altogether. Okay, I do not remember the names of the chromosomes. So one chromosome will have the equivalent of trip A, another chromosome will have the equivalent of trip B. Not that being on the same chromosome will hurt, no. But the fact is, you will make 5 different mRNAs for these 5 genes. So there will be 5 independent transcriptional events. And then each will be translated where the RNA ribosome will be recruited at the 5' end. Unfortunately, an RNA can have only one 5' end. Therefore, in eukaryotes, on the same RNA, you can have only one translation event. There are some exceptions. I will not get into that right now. But generally speaking, the general rule is that in eukaryotes, on one mRNA, you can have only one translation event. It can be several rounds of the same translation event, but only one type. But in prokaryotes, because of the presence of the ribosome binding site on the same messenger RNA, you can have multiple independent translation events and making 5 different proteins from the same mRNA. Now, what is the advantage of having an operon in prokaryotes? That how does prokaryote respond? What is it that the prokaryotes have to deal with? They have to deal with the immediate environment. बाहर tryptophan है कि नहीं है, कम है या ज़्यादा है, is there lactose or not lactose? So depending on that one stimulus, it will decide whether to activate the pathway or not. So it makes sense that if the whole thing is controlled by one unit, then one mRNA is made and all the proteins are made in equimolar proportion, right? Because if there is one mRNA, and then, you know, every little part will have its own ribosome binding sequence and the proteins will be made. So all the proteins will be made in equimolar amount and the event will be done. In eukaryotes, interestingly, the same protein can be involved in multiple different pathways. Therefore, let us say that this one enzyme of tryptophan biosynthesis, I am completely making it up, it is not a fact. Let us say that this tryptophan C gene is also involved in uracil biosynthesis. So when there is a need to make nucleotides in the cell, You do not need to turn on all the other tryptophenyl biosynthetic genes. You only need to turn this on. Therefore, having it in under a different transcriptional control than the other four makes much more sense. So this is the reason why during evolution it evolved such that in eukaryotes you have individual control over genes of the same pathway, while in prokaryotes it is a collective control over genes of the same pathway. Now, when it comes to expression, as someone asked that how do I make an eukaryotic protein in a prokaryotic system? Will I have do I have to engineer the ribosome binding site in insulin? Okay, let us say it is insulin coding. The answer is no. This was the region, where you have the multiple cloning site. Okay, you cloned your thing over here. Right before that, this is a distinction between a cloning vector and an expression vector. In a cloning vector, you will not have this, but in an expression vector, you will have it. So here you will have, depending on what it is, either T7 RNA polymerase binding site or E. coli RNA polymerase binding, E. coli promoter. Okay, it can be either T7 promoter or E. coli promoter. So when you have that, your transcription will start from here, from within the vector sequence. Then somewhere over here will be your ribosome binding site, which is also within the vector. Then it will scan and it will translate from here. You will ensure that you have an AUG right here. That's why it is called an ORF, open reading frame. You only clone from AUG to stop codon, UAA. Okay?
Yes, sir.
So that's how it is done. Any other question? Is it all clear, Akanksha? Did I answer your questions?
Yes, sir.
Okay. Ayush, did I answer your question?
Yes, sir. Yes, sir.
Okay. Any other question?
Sir, I have a question.
Yeah.
Sir, you mentioned that prokaryotes need to respond to that having multiple ribosomal binding sites because they need to respond to immediate surrounding. But wouldn't that be true for unicellular eukaryotes as well?
Yes, it will be true for unicellular eukaryotes as well, but they do have little bit more complexities in life than prokaryotes because they have nucleus. So they have already segregated spatially major cellular events like transcription and translation. Okay, so there are, so like it is not necessary that you will get to see the ramification of it in the unicellular level, but that was the beginning of the, that's why they are very early eukaryotic organisms, but that theme was retained when the next eukaryotic organism evolved because that was beneficial. Okay, see, you need to clearly understand one thing, that none of us should ascribe any intelligence to evolution. Evolution is purely random occurrences. Only those occurrences are selected for which provide survival advantage to the slightly different organism. Okay, so yes, you are right that in unicellular eukaryotes, we cannot perhaps immediately see the benefit of it, but in farther up, it was retained for its beneficial role.
Yes, sir.
Anything else? Okay. Sure, there is no other question.
Hello, sir.
Yeah.
Sir, in multiple cloning sites, sir, it's Kuna.
I can hear you, Kuna. Go ahead.
Sir, in multiple cloning sites, we have multiple restriction enzymes. When we caught the our gene of interest with some restriction enzymes, and we caught the vectors with that same restriction enzyme, when we add to our expression vector, if our desired restriction enzyme which we caught the gene of interest is situated in the middle of the multiple cloning sites, then when we add to the vector and the fragment of gene of interest, then it will change the frame of reference. Then ORF will be changed. Sir,
Okay.
it will affect that.
So I would suggest that you look at it in 2-3 different ways. Okay. Actually, what I made a mistake. So thank you for asking the question. I will do little edits here. I have stopped the share, right? Thank you, Kuna, for asking the question, because I made a mistake and I need to change it here. So, new slide. Can you see it?
Yes, sir.
So, actually what happens is there are 2 restriction sites. One is ND1.
Yes, sir.
Its recognition sequence is CAT-ATG. Okay.
Yes, sir.
Can someone quickly look up the recognition site for this restriction enzyme, NCO1? NCO1.
NCO. I'll Google it.
CCA TGG.
Yeah. These are the only 2 restriction enzymes within which there is an ATG sequence.
Yes, sir.
Okay. So what is done is, thank you so much for asking the question. So if this is your gene, then if this is your RBS, the expression vector will have an ND1 site here, okay, or an NCO1 site here, so that you can, the ATG that you have in your insulin, yes, you will modify it in a manner that when you clone it, from the ribosomal binding site, it will read it like this, starting from this ATG, okay? Because just here I am expanding this region. Okay, so this is your RBS, and then here it is CAT-ATG, and then the rest is insulin sequence. So this ATG came from insulin as well as from your ND1 restriction site. So when the ribosome starts to scan from here, the first ATG it will see is here. So that way the reading frame is set. Okay, you can do the same with NCO1 site as well. Does that answer the question, Kunal?
Sir, like starting from the RBS, hello? Starting from the RBS, like each 3 codon, it will like count to like one, starting from the RBS
so
sequence.
let me quickly search in the net and give you an actual sequence. Okay, yes. Give me a minute. Can you see it here?
Yes,
this is an example of expressing a shrimp lysozyme gene from PET5A vector. PET5A is an expression vector. So this is how the ND1 site is there. Okay, do you see CAT-ATG? So this is where it is cloned, and then this is how it is. So your ribosome binding site will be somewhere over here, and on the, because you are trying to do a expression cloning, so on the 5' side you will not choose any enzyme other than ND1 to begin with. You will have options on the 3' end, but 5' end you will have no option but to use ND1. Does that answer your question, Kuna?
Sir, it answers, sir.
Okay.
Thank you, sir.
Sir, so at the 5' we can use like any enzyme which is having the ATG codon, na?
Yes.
Okay,
and there are only 2 that I know of, NDE1 and NCO1.
Okay.
Yeah. Anything else? See, the advantage is that if you digest with a restriction site, ligate and reconstitute, the restriction site is regenerated, right? So as long as you keep in mind the location of the ATG, you are fine. Yes, Ubaid.
If some inserts, there is no there, sir, in some gene inserts, if there is no this sequence is not present, CAT-ATG, then in that case, what will we do?
No.
If the restriction sequence is not present.
You are mistaken. It is okay. Okay, so let us say that this is the gene that you are trying to clone. Okay.
Yes, sir.
Any gene will have an ORF, right?
Yes, sir.
So there will be an ATG and there will be a UAA. Because you are trying to clone and express the protein, so you know the sequence of it, right?
Okay, sir. Yes, sir.
Yes, sir.
So this is 5', this is 3', this is 5', this is 3'. Now, okay, give me, okay, let me wipe it off. Okay, let me make a larger one. Oh my God. Okay. Next, please supply me some codon names for which you know the amino acids. Look at the table there.
UUU for phenylalanine.
Go ahead. Sorry, you give me all in DNA term. Then whatever, huh? And then let me go to this extreme end. Okay. Let us say now give me stop codon, please. UAA.
Yeah.
Yes,
TAG.
UG.
Huh?
TAG.
TAG. Okay.
Yeah.
Okay. Done. So then this is sorry. Ready? So this is 5 prime, this is 3 prime, this is 3 prime, this is 5 prime. Okay. C T A C C A T, and here C C A C C A C C C A T. Okay, so this is the double stranded DNA sequence. Okay, now what I will do is I will PCR amplify it. Always write 5 prime 3 prime, else I can guarantee that in your class of 28 29 30 no more than 2 will be able to design a PCR primer. Okay, always write 5 prime 3 prime, and remember that DNA elongation happens only from the 5 prime to 3 prime direction. So you want this to be amplified, right? So you want one amplification like a RNA DNA synthesis to happen in this direction and one DNA synthesis to happen in this direction. So when this is your 5 prime, this is your 3 prime, then it has to be complementary to this. So therefore, the 5 prime primer is always exactly the same as the gene sequence that you are trying to do. Okay, and so on. Here it is, you have to think that here it will be this sequence, okay? That is your CTA whatever, CCA whatever. So this is your 5 prime to 3 prime direction, because elongation will happen 3 prime wards. Therefore, you will make your double stranded DNA as the PCR product. Okay. Now what you will do, let us say that just to make my life simpler, this is your 5 prime primer and this is your 3 prime primer. Okay. So primers are synthesized chemically in different facilities, right? All you do is you supply them the sequence of 25, 30 nucleotides and say, please make me this primer. So what you will do then, this is the sequence that you got from your gene, right? This sequence you got from your gene, starting with an ATG. Okay?
Genius.
You can always do this. Can someone give me the sequence of ECOR1 restriction site, recognition site?
GAATTC.
You can add these 9 nucleotides on the 5' end, right? And say that give me the primer, which will be GAATTC, CAT, then ATG, GGT, TTT, blah, 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 blah. Right? And you can say that give me another primer, which will be, give me the sequence of BAM-H1, please. Is it GGATCC?
Yes. Yes, it is GGATCC.
Okay. So then you can always say that give me the 3' primer, like give me this other, like we call it forward primer and reverse primer. Give me the reverse primer sequence, whose sequence will be GGA, TCC, then CTA, CCA, and so on, right? So once you get these 2 primers, they will anneal, the forward primer will anneal. Let me see if I can do this. Nobody wants to make my life easy. So the forward primer will anneal somewhere over here, right? The forward primer will anneal somewhere over here with little bit sequence that is not complementary, which we call the overhang. Overhang has what? Overhang has this ECOR1 site and this CAT, 9 nucleotides overhang. And the reverse primer will anneal somewhere over here. Oops. And it will also have an overhang. Here in the overhang, what you have? You have the BAM-H1 sequence. Okay. Here you have ECOR1 sequence and this little part of ND1. So then when your PCR reaction is complete, then you will have this. This and this, right? Because you artificially added these 2 parts. Now you can clone it in any entry vector, okay? A typical cloning vector. That you put it in a cloning vector because that is your safekeeping. जैसे बैंक में पैसा रखते हैं वैसे. Whenever you want to go for expression purposes, then on this one you will cut this one with ND1 and you will cut this one with BAM-H1. And you will cut your vector, which is like the vector that I am proposing, PET5A, that also you cut with ND1 BAM-H1 and it will go in. Therefore, there is absolutely no need for any such site being in the actual reading frame. Your question should be other way around. In expression cloning, the headache becomes, what if there is an ND1 recognition site here? I have worked with genes where both ND1 and NCO1 site are right in the middle of the gene. What if? Now how would you
Okay,
clone?
sir, that is the problem. Yes, sir.
Then your life is very, very difficult. Okay.
Yes, sir. Thank you, sir.
Okay.
Yes, sir.
Any other question? Piyusha?
Sir, their question are like, I didn't understood about their question that much.
So Ubaid,
So
will you take the responsibility of explaining the whole thing to Piyusha at some other time?
that will be really
Yeah,
nice.
sure,
Actually, the
sir.
last three
sir.
questions I didn't understood much.
Yeah, I will share the recording on the group. Okay.
Yeah.
And then
Okay, sir.
I would ask Ubair to explain the whole thing to you another time. But
Okay, sir.
Piyusha,
Sure,
I
sir.
can tell you one thing.
Yeah.
The only difference is that you have not even heard most of these terms.
No, sir.
I have
That
heard, but I can guarantee none of them knew what it means. I can guarantee that much. So you have to work little bit more. They are not very far ahead of you. I can guarantee you that much. Okay. So do not think that your classmates know significantly more than you. Okay. And you have one advantage which your classmates do not have. There is a possibility that some of them know things wrong because you don't know anything. So chances are that you do not know anything wrong either. Okay. So be brave. Okay.
Yeah.
Anything
Thank
else? Sure. Anything else? Okay. So when it comes to week 0, have we covered everything? Next Monday, we are ready to go to week 1, right? Is genetics?
Yes, sir.
Yes, sir.
Okay, fine. Very good. So enjoy. By the way, the kind of detail with which I explained it to you, I expect you to actually know the subject in that much detail. Okay? Because as I said, that my job is to prepare you for working in a lab. So if you know exactly how primers are designed, what are the different things to think about when you are designing a primer, what is the difference, when you are thinking of expressing in prokaryotes versus eukaryotes. These are the detail to which attention must be paid to. Okay, so I will share the recording, hear them over and over again, but I would fully expect you to learn it. Okay, like I will give you assignments where I will actually ask you to design such complicated things. In fact, I will ask you to design even more complicated things. I have not even started. Okay, so but those are not complicated. Trust me, this is like pure puzzle solving. If you internalize the logic of molecular biology, I will give you few thumb rules in one of these classes. I will give you few thumb rules. If you remember only 6-7 of those rules, the rest of molecular biology you can always understand it starting from those 6 or 7 rules. Okay, you will never go wrong. Okay, chalo. So good night bolte hain?
Okay, sir.
Okay, good night.
Good night, sir.
Good night.
Good night, sir.
Good night.
Good night.
Good night. Good night. Chalo, bye.